Hello and welcome to this video on the exciting and ever-evolving field of neuroscience. Neuroscience is the study of the brain and nervous system, and it encompasses a wide range of topics, from the molecular mechanisms of neuron communication to the complex processes that underlie human behavior and cognition. In this video, we will explore some of the key concepts and recent advancements in neuroscience research. We will take a journey through the brain and its functions, examining topics such as memory, learning, emotions, and sensory perception. We will also discuss some of the latest techniques and technologies that neuroscientists are using to study the brain, from brain imaging to optogenetics. Whether you're a student of neuroscience, a curious individual looking to learn more about the brain, or simply interested in the latest developments in the field, this video is for you. So, sit back, relax, and let's dive into the fascinating world of neuroscience.
Genetics is the study of how traits are passed down from parents to offspring. Each of us has a unique set of genes that determines everything from our eye color to our risk of developing certain diseases. Genes are composed of DNA, which is a long chain of nucleotides. The sequence of nucleotides in a gene determines the amino acid sequence of a protein, which in turn determines its function.
In humans, each cell contains 23 pairs of chromosomes, for a total of 46 chromosomes. Each chromosome contains many genes, which are located at specific locations on the chromosome called loci. Inheritance of genes follows specific patterns, including dominant, recessive, and X-linked inheritance.
Dominant inheritance occurs when a single copy of a gene is enough to cause a trait or disease. For example, the gene for Huntington's disease is dominant, so a child who inherits one copy of the gene from a parent with the disease has a 50% chance of developing the disease.
Recessive inheritance occurs when two copies of a gene are needed to cause a trait or disease. For example, the gene for cystic fibrosis is recessive, so a child who inherits two copies of the gene, one from each parent, will develop the disease.
X-linked inheritance occurs when a gene is located on the X chromosome. Because females have two X chromosomes and males have one X and one Y chromosome, X-linked traits and diseases can affect males and females differently.
Epigenetics refers to changes in gene expression that do not involve changes to the underlying DNA sequence. Epigenetic modifications can be influenced by environmental factors such as diet, stress, and exposure to toxins. These modifications can affect how genes are expressed and can lead to changes in cellular function.
There are several types of epigenetic modifications, including DNA methylation and histone modification. DNA methylation involves the addition of a methyl group to a cytosine nucleotide, which can prevent transcription factors from binding to the gene and decrease gene expression. Histone modification involves the addition or removal of chemical groups from histone proteins, which can alter the way that DNA is packaged in the cell and affect gene expression.
Epigenetic modifications can occur at different times during development and can have long-lasting effects on gene expression. For example, studies have shown that maternal diet during pregnancy can affect the epigenetic profile of the developing fetus and influence their risk of developing certain diseases later in life.
The epigenetic landscape of a cell refers to the various types of epigenetic modifications that can occur, including DNA methylation, histone modification, and non-coding RNA. These modifications can occur at different times during development and can have long-lasting effects on gene expression.
In addition to affecting gene expression, epigenetic modifications can also contribute to the development of complex traits and diseases. For example, studies have shown that epigenetic changes can contribute to the development of cancer, diabetes, and mental illness.
The epigenetic landscape can also be influenced by external factors such as diet and environmental toxins. For example, exposure to certain toxins can lead to changes in DNA methylation patterns and increase the risk of developing cancer or other diseases.
Understanding the epigenetic landscape of a cell is essential for developing personalized treatments and improving patient outcomes. By identifying specific epigenetic modifications that are associated with certain diseases, researchers can develop new therapies that target these modifications and improve patient outcomes.
Epigenetic changes have been linked to a wide range of diseases, including cancer, Alzheimer's disease, and diabetes. In cancer, for example, abnormal epigenetic changes can lead to the inactivation of tumor suppressor genes or the activation of oncogenes. These changes can contribute to the uncontrolled growth and spread of cancer cells. In Alzheimer's disease, epigenetic changes have been linked to the buildup of beta-amyloid plaques in the brain, which are a hallmark of the disease. Similarly, in diabetes, epigenetic changes can affect the expression of genes involved in insulin signaling and glucose metabolism, which can contribute to the development of the disease. By understanding how epigenetic changes contribute to disease development and progression, researchers can identify new targets for therapy and develop new strategies for disease prevention and treatment.
One of the most exciting implications of epigenetics is its potential for personalized medicine. Understanding a patient's epigenetic profile could help tailor treatments to their specific needs. For example, a drug that targets a specific epigenetic modification could be more effective in patients with that modification. Epigenetic biomarkers could also be used for disease diagnosis and prognosis. In cancer, for example, epigenetic biomarkers could help identify patients who are at high risk for recurrence and who may benefit from more aggressive treatment. Similarly, in Alzheimer's disease, epigenetic biomarkers could help identify patients who are at risk for developing the disease before symptoms appear, which could allow for earlier intervention and better outcomes.
While the potential for epigenetics in personalized medicine is exciting, there are also challenges and ethical considerations associated with the field. One of the biggest challenges is the dynamic nature of epigenetic modifications. Epigenetic modifications can vary over time and can be influenced by a wide range of environmental factors, making it difficult to identify a patient's true epigenetic profile. Additionally, there is the potential for unintended consequences associated with the use of epigenetic modifications in therapy. For example, the manipulation of epigenetic modifications could have off-target effects, leading to unintended changes in gene expression. Finally, there are ethical considerations surrounding the use of epigenetic modifications in personalized medicine. Patients may have concerns about the use of their personal genetic and epigenetic data, and the potential for discrimination based on that data.
There is also the need for informed consent, as patients may not fully understand the implications of epigenetic modifications for their health and well-being.
In conclusion, while there are challenges and ethical considerations associated with the field of epigenetics, there is also great potential for new therapies and diagnostic tools based on epigenetic biomarkers. By understanding the role of genetics and epigenetics in human health and disease, researchers can develop personalized treatments and improve patient outcomes.
In addition to the potential for new therapies and diagnostic tools, researchers are also exploring the use of epigenetics in other areas of medicine, such as regenerative medicine. For example, epigenetic modifications could be used to reprogram adult cells into pluripotent stem cells, which could then be used to treat a wide range of diseases.
Another area of research is the development of epigenetic vaccines, which could be used to prevent or treat diseases by inducing epigenetic changes in the immune system. For example, a vaccine could be designed to induce epigenetic changes that increase the expression of certain genes involved in immune function, making the immune system more effective at fighting off infections.
Overall, the field of epigenetics holds great promise for the development of new therapies and diagnostic tools, and researchers are continuing to explore its potential in a wide range of applications.
Understanding the role of genetics and epigenetics in human health and disease is essential for developing personalized treatments and improving patient outcomes. By understanding the epigenetic landscape of individual patients, healthcare professionals can develop targeted therapies that are tailored to their specific needs.
However, it is important to acknowledge the challenges and ethical considerations associated with studying and manipulating epigenetic changes. These include the need for informed consent, the potential for unintended consequences, and the difficulty of identifying a patient's true epigenetic profile. Despite these challenges, the field of epigenetics holds great promise for the development of new therapies and diagnostic tools, and researchers are continuing to explore its potential in a wide range of applications. With further research and development, epigenetics could lead to new treatments for diseases that are currently difficult to treat and could help improve the lives of millions of patients around the world.